Hi, my name is Kate Levinson, and today I'm going to be discussing translation and point mutations. A point mutation occurs when one nucleotide base is substituted for another base. There are two types of point mutations. Transition is when a purine base is substituted for another purine base, or a pyrimidine base is substituted for another pyrimidine base. Transversion is when a purine base is substituted for a pyrimidine base, or a pyrimidine base is substituted for a purine base. The DNA coding strand we are given can be translated into mRNA, which codes for specific amino acids, as seen. The UGA codon codes for the stop amino acid, so we don't continue reading the mRNA after. Here is our original information. Let's begin with the first point mutation. Our first point mutation is the missense mutation. This changes the amino acid and can alter the protein function. In the mutated DNA coding strand, we can see that the C is what changes, and in the original DNA, it was a T. When it is translated into mRNA, it stays a C, but in the original mRNA, it was a U. So the original amino acid is tryptophan, but in the mutated amino acid, it is arginine. This is transitioned from T to C, so it's a pyrimidine to a pyrimidine. The second mutation that we have is the nonsense mutation, and this is when a codon is changed to a stop codon. In our mutated DNA coding strand, we see that the A is where the mutation is, and in the original DNA strand, it is a G. When it is translated into mRNA, it becomes UGA, but in the original, it was UGG, which codes for tryptophan. But in the mutated amino acids, it turns into a stop codon, so we don't read any more after. This is a transition from G to A, so it is a purine to a purine. The next mutation is the silent mutation. This is when the mutation codes for the same amino acid. In the mutated DNA coding strand, we can see that the mutation is at the A, so it's GCA. But originally, it was GCT, so it's mutating from a T to an A. When we translate this into mRNA, it changes from GCU, which codes for alanine, into GCA, but it still codes for alanine. This is transversion from T to A, so it's pyrimidine to purine. The last point mutation is the readthrough mutation, and this is when a stop codon is changed to a codon that codes for an amino acid, so we will keep coding the mRNA. In the mutated DNA coding strand, we see that it is AGA, but in the original it is TGA, which codes for UGA. UGA is a stop codon. In the mutated, AGA translates to AGA, which codes for arginine, which is not a stop codon, so we will keep reading the mRNA. The next two amino acids are proline and glycine. This is a transversion from T to A, so it is a pyrimidine to a purine.